Hello students, welcome back to Zoology online class. Today we are going to discuss about sex determination in human and mutation. Lesson number 18. So listen carefully. Sex determination in human. This is a human cell. Here you may see some thread-like structures in it. These thread-like structures are known as chromosomes. Thread-like structures are known as chromosomes. There are 23 pairs or 46 chromosomes in a human cell. Human cell is having 23 pairs of chromosomes. Out of this, 22 pairs or 44 chromosomes are autosomes. So first 22 pairs of chromosomes are known as autosomes and one pair is called sex chromosome. The last pair, 23rd pair is called sex chromosome. These sex chromosomes carry instructions called sex determination. Okay, but how? That only we have to see. So sex chromosomes carry sex determination. How? Let's see. Sex chromosomes are of two types. Sex chromosomes are of two types. They are X chromosome and Y chromosome. X chromosome and Y chromosome. In a female, only one type of sex chromosomes are there. Okay, in a female, only one type of sex chromosome, which is X chromosome. Okay, so 22 pairs of autosomes and one pair XX. That is a sex chromosome. But in a male, XY sex chromosome is present. XY, 22 autosomes plus XY chromosome is present. Let us see the arrangement of chromosomes in an unfertilized egg. In an unfertilized egg has 22 autosomes and one X chromosome. Unfertilized egg, 22 autosomes are present plus only one X chromosome is present. Whereas sperm have two types of arrangement. This is unfertilized egg, okay, female. So 22 pairs of autosomes and one X chromosome. And in male, the sperm is having two types of arrangement. One has 22 autosomes and one X chromosome. One has 22 autosomes plus one X chromosome. In another arrangement, okay, but another type has 22 autosomes and one Y chromosome. So two types of arrangement. Here X, there Y. Okay, well, sometimes maybe X, sometimes maybe Y. Now see how the sex of a baby is determined in man. Okay, so when an egg is fertilized by a sperm containing X chromosome, the zygote formed would have two XX chromosomes. Okay, two XX chromosome. This zygote is developed into a female child. XX means that is a female. Okay, female child. So this zygote developed into a female child. When an egg is fertilized by a sperm containing Y chromosome, the zygote formed would develop into a male child. Okay, XY, that is a male child. Understood? XX means that is a female, XY that is a male. So, the father's sex chromosome that decides the sex of a baby. Okay, so the father's sex chromosome only decides the sex of a baby. Maybe that is a male child or female child. Mutation. The term mutation was introduced by Hugo de Vries in 1901. The term was introduced by Hugo de Vries. So first let's see what is mutation. Changes in the nucleotide sequence of the DNA. Mutation is an inheritable sudden change in the genetic material or DNA of an organism. A mutation is a change that occurs in our DNA sequence either due to the mistakes when the DNA is copied or as the environmental factors like UV light, X-ray, etc. Sometimes the DNA is copied at that time. There is a mistake in the DNA sequence or the environmental factors like UV light, X-ray, etc. The process by which mutation is produced is called as mutagenesis. The process of mutation that is known as mutagenesis. The chemical or physical factors or agent that increases the rate of mutation is called as mutagen. Some of the factors increases the rate of mutation. That is known as what? Mutagen. So, in this sequence you see this is a original DNA sequence. Here there is a change, there is a sudden change in this area. So, mutated sequence, original sequence and mutated sequence. So, we can see the difference. The next one, general characteristics of mutation. Generally, mutation takes place during the meiosis of cell division. During the cell division only this mutation takes place, that is meiosis cell division. Mutations are recessive. Mutations are generally harmful to the organisms. Mutations are random, occur at any time in any cell of the organism. The same mutations may occur again and again. These are the general characteristics of mutation. The next one, role of mutation. This mutation is the source of genetic variation. It is the raw material for evolution. It generates a phenotype differs from the normal one. See here, original sequence, and here there is a mutant copy. There is a change. The next one is chromosomal mutations. Chromosomal mutations. So what is chromosomal mutation? The sudden change in the structure or number of chromosome is called chromosomal mutation. There is a change in the structure or number of chromosomes. That is known as what? Chromosomal mutation. This chromosomal mutation, two types. Changes in the structure of chromosome, and another one, changes in the number of chromosome. Changes in the structure of chromosome and changes in the number of chromosome. First, changes in the structure of chromosome. Structural changes in the chromosomes usually occurs due to errors in the cell division. So this type of chromosome changes during the cell division only takes place. It leads to deletion, duplication, inversion and translocation. So four types: deletion, duplication, inversion and translocation. First one, deletion. Due to breakage, a piece of chromosome is lost. See this is a chromosome, is it? So breaks in the chromosome. So one part breaks. So here the deleted genetic material. So the particular area removed. So here remaining parts joined. So this is known as deletion. A piece of a chromosome is lost. This is the first type. Second one, duplication. This duplication occurs when a gene sequence is repeated. See, this is a normal chromosome. Here in this area repeated. This area repeated. So two times it is present. A section of DNA is duplicated. So this is known as duplication. Occurs when a gene sequence is repeated. That is duplication. Then third type is inversion. Chromosome segment breaks off, segment flips around, backward segment reattaches. So this area, see here, breaks in the chromosome, this area. So in this second figure, that particular area reinserted upside down. Okay, in the third one, see, just upside down. Okay, so this is known as what? Inversion. Segment flips around, backward segment reattaches. That is inversion. Then fourth one is translocation. Fourth one, translocation, involves two chromosomes that are in homologous. Here two chromosomes needed, but both are different. Now that are in homologous, so that are different. Both the chromosomes are different. Part of the chromosome is transferred to another chromosome. So in this part is transferred into another chromosome. Okay, see the after translocation, just you see the difference. So this area transferred into this chromosome. So this is known as what? Translocation. Second type is changes in chromosome number. Each organism contains a characteristic number of chromosomes in its cells and gametes, that we all know. Male and female gametes are haploid. Male gamete is a sperm, female gamete is a egg. They are haploid. N. They contain basic set of chromosome or single genome. During fertilization, male and female gametes fuse and form zygote. So during the fertilization, the sperm and egg fused together and form zygote. That is, the zygote is diploid. 2N. It contains two genomes, one from father and another from mother. Zygote undergoes embryological development and forms an adult. So the zygote developed into an adult. Sometimes an abnormality occurs during nuclear division or due to presence of chemicals or radiations. Changes in chromosome number takes place. Sometimes the chromosome number, there is a changes in the chromosome number takes place. So sometimes during the nuclear division or some of the chemicals, presence of chemicals, radiations, because of that the chromosome number changed. Okay, there is a change in the chromosome number takes place. The next have to see types of changes. Changes in number of whole chromosome is called heteroploidy. Changes in number of whole chromosome, that is known as what? Heteroploidy. This heteroploidy is divided into euploidy and aneuploidy. First one, euploidy. Euploidy means change in entire set of chromosome. There is a change in the whole set of chromosome, that is known as what? Euploidy. So heteroploidy divides into two, euploidy and aneuploidy. Aneuploidy means loss or addition of single chromosomes. That is a loss, sometimes deletion, sometimes addition of single chromosome. Okay, first have to see euploidy. What is euploidy? Euploidy refers to genomes which contain chromosomes that are multiple of some basic number. Basic number, that means X or N. Sometimes this X is multiplied. Okay, some X or N is multiplied. 3N, 4N, 5N, like that it is multiplied. Okay, individuals containing the basic set or number of chromosomes are called monoploid. Basic set means that is X or N. That is known as monoploid or haploid, basic set. Okay, multiplication, that is known as what? Euploidy. Okay, then similarly 2x or 2n diploid. The 2n condition is known as diploid, 3n condition is triploid, 4n is tetraploid, 5n is pentaploid and 6n is hexaploid. Okay children, but this 2n is common, that is normal. Diploid condition is normal in all the organisms. So the remaining this 4, that is known as what? Polyploids. Remaining 4 is known as polyploids. Aneuploidy. Aneuploidy means loss or gain of a chromosome. Sometimes there may be addition of chromosome, sometimes deletion of chromosome. Aneuploidy has two types, hypoploidy, hyperploidy. Hypoploidy means loss of chromosome, hyperploidy gain of chromosome, deletion and addition. Hypoploidy, loss of one or more chromosome, deletion of one chromosome or more than one chromosome. It is of two types. First one, monosomy. 2n minus 1. It is due to loss of a single chromosome. So this monosomy takes place because of the deletion of single chromosome. They produce two types of gametes, one with n chromosome and another with n minus 1 chromosome. Monosomy causes lethality in diploids but has no effect in polyploids. Lethality means the capacity to cause death. So in diploid condition it leads to death, but polyploid condition there is no effect. Second type, nullisomy, 2n minus 2, loss of a pair of chromosomes. So deletion of 2 chromosomes. It causes lethality. Nullisomy causes lethality. The next one, hyperploidy, gain of one or more chromosomes. Gain of addition of one chromosome or more chromosomes. So first type, trisomy, 2n plus 1, gain of a single chromosome. So addition of one chromosome. In humans, trisomy causes 3 types of syndromes. This trisomy causes 3 types of syndromes. First one, Down syndrome. It is caused by trisomy of 21st chromosome. There is a change in the 21st chromosome or addition of chromosome in the 21st. So 21st one more chromosome is added. It occurs in 1 by 650 births. It is also called Mongolism. The children are mentally retarded, large tongue, small ears, short stature and enlarged liver and spleen. So these are the symptoms of Down syndrome. Mentally retarded children, they have large tongue, small ears, short stature and enlarged liver and spleen. The second disease, Edwards syndrome. It is caused by trisomy of 18th chromosome. That is the addition of chromosome in the 18th pair. It occurs in 0.3 per 1000 births. The symptoms are mental retardation, cardiac malformations and deformity in skull, face and feet, hair lip and cleft palate often occurs. Death occurs around 3 to 4 months. So anyway, at last death only. Then Patau syndrome. Trisomy of 13th chromosome. So addition of chromosome in the 13th pair. It occurs in 0.2 per 1000 births. The symptoms are mentally retardation, hands and feet are deformed, heart and kidney defects, death occurs within hours or days of birth. So hyperploidy, trisomy. So trisomy produces the 3 types of syndromes, but in your book only one syndrome is given, Down syndrome. So remaining just for your remembrance only I told. See this arrangement of chromosomes. So here totally 23 pairs of chromosomes are present, 22 autosomes and 1 pair is known as allosome. In this 21, chromosome number 21, here one extra chromosome is present. Is it? 3 chromosomes are present in this area. So totally 47 chromosomes are present. So that condition is known as what? Down syndrome or trisomy 21. Trisomy 21. In the 21st pair, 3 chromosomes are present. Okay. So it is associated with this problem. This is associated with mental retardation, delayed development, behavioral problems, weak muscle tone, vision and bearing disability or some of the conditions seen in these children. Okay, see this is in the early stages of development. So here lot of problems are there. When you see itself we can find out, is it? So these persons are adult persons. Anyway, just when we are looking itself we can identify they have some other problems. So this condition is known as what? Down syndrome. Gene or point mutation. Definition. A point mutation is the type of mutation in which a change in single or few nucleotide at a single location of DNA sequence. So this point mutation is a type of mutation and there is a change in the DNA sequence at a single location. In only one place only there is a change. That is point mutation. Then types of point mutation. There are 2 types of point mutation, base substitution and frameshift mutation. Base substitution and frameshift mutation. So first let's see what is base substitution. What is base substitution? So this base substitution occurs when one base is switched out with another base. So one base is removed, instead of the particular base another one base is inserted. That is base substitution. So this is a normal DNA sequence A, T, G and C, T, C. Okay, so here in this sequence here A, T, G and A, T, C. So instead of this C, A is inserted here. Instead of the C, A is inserted. So this is substitution, base substitution. So one base is switched out with another base. One base is removed and another one base is inserted. That is known as what? Base substitution. Then the second type is frameshift mutation. Frameshift mutation. This frameshift mutation occurs when one base is added or removed. So one base is added or removed. Say here ATG, CTC. So in between these two, one base is inserted, one base is added. So this is known as what? Insertion. One base is added. Here deletion ATG, TC. That C is removed. So this is deletion. So frameshift mutation occurs when one base is added or removed. These are the 2 types, insertion and deletion. Textbook evaluation. Roman number 1, choose the correct answer. Question number 7. The number of chromosomes found in human beings are 22 pairs of autosomes and 1 pair of allosomes. 22 autosomes and 1 allosome. 46 autosomes, 46 pairs autosomes and 1 pair of allosomes. Answer A. 22 pairs of autosomes and 1 pair of allosomes. Question number 8. The loss of one or more chromosome in a ploidy is called dash. Tetraploidy, aneuploidy, euploidy, polyploidy. Answer B. Aneuploidy. Roman number 2, fill in the blanks. Question number 5. An inheritable change in the amount or the structure of a gene or a chromosome is called dash. Mutation. Answer mutation. Roman number 3, question number 7. Roman number 3, question number 7. That is a true or false. Down syndrome is the genetic condition with 45 chromosomes. This sentence is false. Correct answer 47. Down syndrome is the genetic condition with 47 chromosomes. Roman number 4. Match the following. So here also only the correct answer is given. Autosomes 22 pair of chromosomes. Diploid condition 2n. Allosome 23rd pair of chromosome. Down syndrome trisomy 21. Dihybrid ratio 9:3:3:1. Roman number 7. Question number 3. The sex of the newborn child is a matter of chance and neither of the parents may be considered responsible for it. What would be the possible fusion of gametes to determine the sex of the child? Answer page number 269. Page number 269. Sex determination in human. Under that first line. Human beings have. Start from that and same page right side till the end of the first paragraph. Then Roman number 8. Question number 3. Kavita gave birth to a female baby. Her family members say that she can give birth to only female babies because of her family history. Is the statement given by her family members true? Justify your answer. The statement given by her family member were not true. It is not hereditary or family history. The sex determination is mainly depends on which category of sperm fuses with the egg. If the egg is fused by the X-bearing sperm, an XX individual, that is female, is produced. If the egg is fused by the Y-bearing sperm, XY, male individual, is produced. So, the sex of the baby depends on the type of sperm which fertilizes the ovum.